All right then, let's talk about how does an impulse pass along a nerve fiber. So basically we'll be talking about how this action potential is produced. Okay then, so the first step here is, so first that you see here is that the polarized nerve fiber with the resting potential. So this is actually the nerve or the axon or the neuron actually at resting potential level, right? So this is this part here. This is straight horizontal line that you see at minus 70 millivolts. Okay, this is the resting potential, and this is this axon at rest right now. Okay, then you see there comes a stimulus. Okay, so this stimulus can either generate an action potential, right? So you can see this curve here, or there might be a few failed initiations. So these failed initiations means that this threshold value actually has not been reached. So the resting potential is minus 70 millivolt and the threshold is minus 55 millivolt, right? So till the time this threshold is reached, only then an action potential will be generated, right? If the impulses are the stimulus is weak enough, so it doesn't generate this much energy that actually it would pass this threshold value. So these nerve impulses would not be generated at all. So they would die here and right now here, right? But so we need to pass this threshold of minus 55 millivolt, only then a nerve impulse will be conducted, right? Then you can see this along the x-axis, look at the time. So this is the time in milliseconds. So you can see how quickly it passes actually, right? So for a stimulus to get generated, right, and to reach the peak, so you can see it happens in around 1 milliseconds only, right? So the stimulus, if it crosses this threshold of minus 55 millivolt, right, and so this phase here actually will reach in 1 milliseconds only, right? Then the second phase also, it goes from 2 till 3, again it takes 1 milliseconds, right? So you can see how quickly an action potential actually occurs. Okay, so the first stage here was this resting potential, the axon at rest, right? Then comes this generation of action potential and then propagation of action potential. This is the stage that we call as depolarization, right? So depolarization. This is the part where actually these voltage gated sodium channels open, right? Because as we discussed earlier, these voltage gated channels remain closed, right? So the moment they open is the time when sodium ions actually start getting in. Because if you can recall from the resting potential, the sodium potassium pump was actually pumping out 3 sodium ions out, and then it was bringing in 2 potassium ions in. But what is happening during an action potential? These voltage gated sodium channels open, and then they allow these sodium ions actually to enter the cell, right? So this is the phase that we call as depolarization, right? So that's this stage here, right? And it reaches when the threshold value actually is crossed, right? And then you can see it peaks at around plus 40. So in some books you would find plus 30. In fact, in our book it is written as minus plus 30 millivolt, right? So you can see from negative, look at the value now. It has actually turned positive now. So much positive ions actually they enter inside the cell. So much that it reaches plus 30 millivolts. So again, I was saying in some books you would find the value as plus 40 millivolts. In some book you would find as plus 30 millivolts. So it's better to go according to your book, but it never ever actually peaks above this value. So this is the fixed value, right? So it would go this high only. So when this happens, when it reaches plus 40 millivolts, now is the time when these sodium channels close. These voltage gated sodium channels close, and now is the time when the voltage gated potassium channels would open, right? And now since the environment inside is positive, so what would happen? The potassium ions would start to go out because of those voltage gated potassium ions. And now you look at the curve here. So you see it is coming down from plus 40, so much so that it even crosses this value of minus 70. So much positive ions actually go out that actually this value can go past like minus 80 millivolt or minus 85 millivolt. So it even becomes more negative now because more positive charges have actually gone out, right? Until this time, the sodium channels are kept tight shut. right? So these more potassium ions actually go out during this phase. So this is the phase that we call as hyperpolarization, right? Because more positive ions actually have gone out, and then inside of the cell actually has become more negative now. So this is a phase which we call as hyperpolarization, right? Okay, now is this phase as you're looking at here. So this is actually a little recovery phase, which we call as a refractory period, and it takes around 1 millisecond. So you can see here from this like around 3 to 4 milliseconds, it takes around 1 millisecond to get back to the resting state. So this is the time when the potassium channels would close. So no more potassium ions would go out. And now is the time when the sodium channels would open, right? And it would actually bring it back to minus 70 millivolts. So the resting potential will actually be maintained then. So this is how, just an overview of how action potential is created. So we start off with the resting potential, which is minus 70 millivolt. If the stimulus is high enough, right? So the first stage will be depolarization, when the voltage gated sodium channels open and they allow sodium ions to go in. And this is actually a positive feedback. So we'll just talk about that in a while. So actually, once like some sodium ions get in, and like actually after that, some more channel sodium ions get in. So this depolarization happens till the time this value reaches plus 30 millivolts. And after that, these voltage gated sodium channels would close and potassium channels would open, right? These voltage gated potassium channels would open. So what would happen, because the internal environment now has become more positive, so now the potassium ions would go out. And since these are also positive, so as they go out, right? So these potassium channels, they remain open, okay? And then this value can reach to minus 80 millivolt. Only then these potassium channels would close and then the sodium channels would open. And this would bring it back to the resting state of minus 70 millivolts. Okay, so that was just an overview. We will talk about every single stage in detail now. So first is the polarized nerve fiber, which is actually at the resting potential value of minus 70 millivolts. So the reason for that was actually that the outside of the inactive nerve fiber when it is at rest, right, it is positively charged because just from a discussion you might remember there were more sodium ions outside. So the outside of the nerve is more positive compared to the inside, which is negative as compared to the outside, right? So that's the reason the nerve fiber is actually polarized and the value is kept at around minus 60 millivolts to minus 70 millivolts. As I just said, you might find these values in the books. So stick to the value that is given in your book, right? So the resting potential value stays at this value. So that is step one, which is axon at rest. So you see here, these are the voltage gated channels. So they are closed. So this is a sodium channel, voltage gated sodium channel, so it's closed. See, and look at this one. This is a potassium channel, voltage gated channel, which is also closed, right? This is the sodium potassium pump, which is actually working hard right now to maintain this resting potential, right? And as we just discussed, it sends 3 sodium ions out, and at the same time brings in 2 potassium ions in. So this is actually the resting potential kept at around minus 70 millivolts, right? Okay, so step number 2 is the generation of action potential. So when a stimulus is given and it is strong enough, the permeability of neurolemma is simply the cell surface membrane of a neuron is known as neurolemma. So this permeability changes because those voltage gated sodium channels open, right? And as the sodium ions move inside, this causes depolarization, right? And because of that, what happens, like at the initial stage, because the resting potential was minus 70 millivolts, so this actually reaches to around minus 55 to minus 60 millivolts. So it reaches a threshold value first, right? So like a small number of sodium channels open. So a little sodium ion actually gets in. So this value actually increases from minus 70 millivolts to minus 55 millivolts or minus 60 millivolts, right? And after that, if it like more enters, so more channels would open, right? And then all the channels would open and sodium ions would rush into the axon. And at this point, actually, complete depolarization will take place. So if you recall from this part here, so complete depolarization will take place at this point here. right? So all of the voltage gated sodium ions will open eventually one by one, right? And we will reach this action potential. And once it reaches this polarity of plus 30 millivolts, right? So after what would happen, these voltage gated sodium channels would close. So if these channels are closed, so this sodium would actually be trapped inside now. See, since the sodium is trapped inside, so this is what is actually causing a more positive environment inside now, right, as compared to outside. Got it? Because the sodium is now trapped, it cannot go out. Okay, so let's talk about these action potentials in detail now. So there are channel proteins, as we were just discussing. So these are channel proteins in these axon membrane. So these channel proteins, okay, there are of two types for sodium as well as for potassium, right? So in this axon membrane, so there are channel proteins for sodium and channel proteins for potassium ions to pass through. So these actually open or close depending upon the electrical potential across the axon membrane. So that's the reason they're known as voltage gated channel proteins, right? And as we just discussed, during resting potential, when the axon is at rest, these channels remain closed. Got it? So when an action potential is stimulated by a receptor cell in a neuron, the following steps occur. So step by step, let's just learn them. So what happens first, the voltage gated channel proteins in the axon membrane open, right? So at step number one, depolarization will happen. So sodium ions will pass into the axon. So it's the sodium channels that actually open first. Sodium ions pass into the axon down the electrochemical gradient because there were more sodium outside, right? So there was a steep concentration gradient, and since the inside of the cell was negatively charged, so already positive charges are actually attracted towards them. So these sodium ions pass into the axon down the electrochemical gradient, right? And actually this causes the movement of sodium ions inside the cell now. Now, what happens because of that? This reduces the potential difference across the membrane and the inside of the axon becomes less negative. So remember that value plus 30 millivolts. So this is what is known as depolarization, right? So what happens because of that? More channels are open. So this triggers more channels to open. And thus this allows more sodium ions to enter, right? And it causes even more depolarization. So it happens step by step. So that value of plus 30 millivolts is not reached in one go, right? So it keeps on increasing little by little. So this triggers more channels to open, it causes more sodium ions to enter, causes more depolarization. So this is actually an example of positive feedback, right? So why we call it a positive feedback? Because a small initial depolarization leads to greater and greater levels of depolarization. So I hope you're getting the concept here, right? Okay then. Let's talk about the threshold value also. So if the potential difference reaches around minus 50 millivolts, so remember it started off with minus 70, right? So minus 70 millivolt was the resting potential, right? So this is how it started. It was at rest at this time. So by the time the potential difference reaches minus 50 millivolt, which is known as the threshold value, right? More channels would open, right? Because we need to actually attain this value. If this value is not reached, channels would not open, right? So many more channels open, many more sodium ions would enter. And then because of that, what would happen? Because more positive charges are coming in, it actually increases this potential to plus 30 millivolts. And remember, this value is fixed, right? In some books, you would find around plus 40 millivolts, but it does not go beyond that. So have a look here. So this is the stage of depolarization where these voltage gated sodium channels are open. See, so only these are open. These voltage gated potassium channels are closed at this time. So now since these are open, so what would they do now? So they would actually now bring in three sodium, like they would bring in these sodium ions inside. So this will keep on increasing and increasing and increasing. So at the initial moment, we had only a few sodium ions inside. So the opening of these voltage gated channels would increase the inflow of sodium ions inside till the time it reaches plus 30 millivolts, right? So here you can see this voltmeter also. So it is actually showing at plus 30 millivolts. So this is what we call as depolarization, right? And if the threshold is reached, more and more sodium like channels would open and they will actually keep on bringing in the sodium inside. Okay, so this action potential actually creates a positively charged area inside the axon as we were just discussing. And because of that, a local circuit is generated between the action and the resting potential areas. So if you look at this part, so there's a strong stimulus is actually acting at, actually this is how this action potential is being propagated, right? So more and more channels would open eventually. So positive feedback will be created, right? And this is how an action potential will be propagated. So it will actually act as local circuits. So it will actually act as a local circuit between the action and the resting potential areas. We just saw that in the diagram. So what would happen because of this, the selective permeability of the resting region would change, right? And this action potential will be propagated along the nerve fiber after initiating it. Okay, so that's the reason we see an impulse in the form of a wave. Okay, so we see a wave of action potential which propagates along the nerve fiber due to depolarization. So this wave here that we are looking at, this is how this action potential is being propagated. This one, the depolarization. Okay, then so after depolarization, the next stage is repolarization. Right, so this happens around after 1 millisecond, after an action potential has been generated, right? So then it starts the repolarization stage. So this is the time when all the sodium voltage gated channels close, right? So repolarization happens when all these channels close, the sodium voltage gated sodium channels that were open, that were letting the sodium in and which actually increase this membrane potential till 30 millivolts, right? So these channels would close. And because of their closure, no further sodium ions can actually diffuse into the axon now. This is the time when potassium voltage gated channels would open and they would actually allow these potassium ions out of the axon because now like this potential actually has to be taken back towards the resting potential, right? The action potential has already been generated, right? So now potassium ions, they are also positively charged, remember, so they are actually now pumped out of the axon. And again, this is done down the concentration gradient because remember, we had more potassium ions inside, right? So they are actually pumped out. So because of that, the potential difference, which actually had reached to plus 30 millivolts, it actually uh, like like what do you say? So this returns the potential difference actually to normal, and which is actually the same plus 70 millivolts. So this whole process is known as repolarization. Yeah, so we are talking about this phase. See, so the action potential was reached. So after about 1 millisecond, so see if this action potential reached at around 2 milliseconds. See, so from 2 till 3. So it takes around 1 millisecond. So this repolarization will happen and all the sodium channels would close and the potassium channels would open. These potassium channels, as they open, they would actually diffuse out all these potassium ions, which are actually themselves positively charged. They will go out, right? And this will actually try to bring down the membrane potential back to minus 70, right? So this is repolarization, okay? But you see here, actually more potassium goes out, right? So this, like the membrane potential actually drops down even to minus 80 or minus 85 millivolts, right? So these potassium ions, these channels, they remain open, right? And this part here is what we call as hyperpolarization. So more potassium ions, more positive charges actually go out. And because of that, the inside of the cell, this interior becomes more negative now. So this is a brief period of hyperpolarization. So this is a brief period when the potential difference across the section of the membrane becomes more negative than the normal resting potential. And what happens now, potassium voltage channels would now close. And as they close, the sodium channels actually, now they will become responsive to depolarization again. So they are kept tight shut, right? And once these potassium channels close, only then these sodium channels become responsive again. Until this occurs, okay? So that's the period of recovery. And this is what we call as this unresponsive phase. And after this hyperpolarization, this phase, until the time these sodium channels open, this is known as a refractory period. So this takes around only 1 millisecond, but this is what we call as a refractory period. So by the time these sodium channels become responsive again. So what we call as a refractory period. So let's have a look at this hyperpolarization once again. So now you see these sodium channels, voltage gated channels are closed now, and these potassium voltage gated channels are open now, right? So what you see, we have more sodium inside, because as this channel was open, so it brought in a lot of sodium ion inside. So we have more positive charges inside. So it has reached to plus 30 millivolt. And now after closure of these voltage gated channels, the potassium channels open, and now they take these potassium ions out, right? And a lot of them will actually go out, right? And if you see, actually, it can drop down to around minus 85 to minus 90, right? So that would be hyperpolarization, right? So in repolarization, potassium ions are going out, but more of it actually goes out because these channels remain open for a little longer, right? So more positive ions go out, and it actually drops this membrane potential to even minus 85 or minus 90 millivolt. Okay, so a while back we were talking about the refractory period. So just remember, like it happens like only for about 1 millisecond. So once this resting potential is close to being re-established, like because it has dropped down to around minus 80 to minus 90, right? And it is close to be re-established. So what happens? This potassium voltage gated channels would now close, and this is the time when these sodium ions, these channels would actually now open, and they would become more responsive to depolarization again. But until this occurs, until this recovery occurs, right? So this is what is known as a refractory period. So it takes around 1 millisecond. Now, these refractory periods are very, very important because of the following reasons. Now, this refractory period, like this period where these sodium channels actually become responsive, it ensures that action potentials are discrete events, right? So they do not merge. So they're separate events. They do not merge. So it actually ensures that these are one event at a time, right? So it stops them from merging into one another. It also ensures that new action potentials are generated ahead, right? So action potential travels ahead, like say it doesn't go back. So this is why these sodium channels are remained tightly shut. They are closed. So if they are closed, so no action potential can be generated till the time they're closed. So these refractory period, they ensures that new action potentials are generated ahead. It means further along the axon rather than behind the original action potential, right? Because the region behind, is actually recovering right now because action potential has already occurred there. Okay? This means that this impulse can only travel in one direction, which is very, very essential for a successful and efficient transmission of a nerve impulse along the neurons, because you eventually want a response, right? And for to getting a response, we need this impulse to travel in only one direction, right? And this also means that there is a minimum time between action potentials occurring at any one place along a neuron, right? So you just saw, okay, this happened in around like 5 milliseconds, right? The whole process from start till the end, right? So the length of the refractory period is actually the key in determining the maximum frequency at which impulses can be transmitted along the neurons. So usually it can take around like 500 to 1000 per second, right? So these many impulses can be generated, right? So this length of the refractory period is actually very, very important in that case. It's a key. Okay? So if you want to achieve maximum frequency, so this length of refractory period is actually of utmost importance in this case. So remember, during this refractive phase, only a section of the axon is unresponsive. Okay, so remember that. It's just a section of axon is unresponsive. So it actually means that new action potential is generated ahead, right? Okay, so just to recap the whole process. So this is how it started. Resting potential was kept as minus 70, right? So this resting potential was actually maintained because of these sodium potassium pumps, remember, right? So what was this pump trying to do? It was trying to send the sodium out. At the same time, it was bringing potassium in. So it was keeping this resting potential at minus 70 millivolts, right? The second phase is depolarization, when these voltage gated sodium channels open. So you can see here, right? The voltage gated sodium channels open. So this yellow one that you see here. So now the sodium is actually getting in. And this is what causes this depolarization. So this threshold is met. So once it crosses, so this is how the action potential will be propagated. This is how depolarization till this time, these voltage gated sodium channels would remain open, right? And it will like stepwise it would happen. So giving it a positive as a positive feedback, right? Then comes repolarization, which is the time when these orange, these potassium channels, actually voltage gated channels, now they open and they allow this potassium to go out, right? So now you see from plus 30 millivolts, the potential membrane potential actually coming down now. So this is repolarization. So till the time voltage gated potassium channels are open and potassium is actually now going out, right? But by the time these channels remain open, so more potassium actually shoots out, right? So it can drop down to around minus 80, minus 85 millivolts, right? Then comes this refractory phase, right? Because this recovery phase, it takes a little while for these sodium pumps to open again, right? And then again, it brings come to this resting potential. So by this time again, the sodium potassium pump start to work now because remember, like it had to bring three sodium ions out. So since already more negativity has been created here, right? So now these pump will start to work to bring it back to minus 70 millivolts. So this is another diagrammatic view. So just have a look how it looks like. So you see here, so here they have shown you a motor neuron and look at the direction of the action potential, right? So it will travel along the axon till the time it reaches this, the nerve endings here, right? So this is how it starts. So in response to a signal, the soma, and soma simply means the cell body. So this end of the axon becomes depolarized. So now you can see the charges. So we have more positive charges inside now. So this part of the axon, because as we just discussed, it happens stepwise, right? So this part of the axon becomes depolarized. So we have more positive charges inside now, right? And what happens now, this depolarization then would actually like move on to the next part. But by this time, after the depolarization, the next step is repolarization. So this part actually will repolarize. So by this time, the sodium channels would close, potassium channels would open. But at the same time, you can see this depolarization has actually moved to the next part. So this actually how this action potential is being propagated. Clear? And then you see this part here is now resting. The next part here is now repolarized. So look at how this is being propagated. The next part here is not depolarized. So this is how this action potential continues to travel down the axon. So look at the direction, only goes in one direction, right? So look here. In repolarization, like since potassium ions are going out, so it is becoming negative again, right? And then this is the refractory phase, right? And then look at depolarization again, more positive charges inside now. So this axon potential at any point in an axon cell surface membrane triggers the production of an action potential in the membrane on either side of it. So when we say on either side of it, so it means actually this movement of positive charges inside, right, from outside. So this temporary depolarization of the membrane at the site of the action potential causes a local circuit because now the positive charges have moved in. So this local circuit sets up between the depolarized region and the resting regions, right, on either side of it, because the part above it is also at rest and the part behind is also at rest, right? So it is localized. So you can look at this diagram also. Okay, so this is a localized part. Then it happens here next to it. Then it happens here. Okay, so these local circuits depolarize the adjoining regions and so generate an action potential in them, right? So usually what happens is usually happens in experimental situation when a stimulus is applied somewhere along an axon. So in practice, like if you want to do an experiment and if you want to look at how this action potential generates, so this can be done in an experimental situation also. Okay, so you can do that in an experimental situation, but when it comes to the body, the human body, these action potentials, they begin at one end, okay, and then new action potentials they're generated ahead. They're never generated behind it. So this is what actually happens in a human body or generally in any living body, right? So this is because the region behind it, as we just discussed, is still recovering from the action potential because it has had already one action potential. So it is still recovering from it, right? And the sodium ion voltage channels that we just discussed, they remain tightly shut, right? So they cannot be stimulated to open at all. Okay? Even though if the stimulus is so great, even then they will not open. So that's the reason. This is why this is known as a period of recovery or the refractory phase. And this is the time when the axon will remain unresponsive, right? And there are several consequences of there being refractory periods. So as we just discussed, action potentials are discrete events. Remember, they do not merge, right? And then we discussed about the time also. There's a very minimum time between two action potentials occurring at any one place. Remember, times is in milliseconds, right? And the length of the refractory period determines the maximum frequency at which the impulses are transmitted along the neurons, right? So these are the consequences that why is there a refractory period. So this is another view. Here, this is the resting phase. So at resting phase, you see this is the sodium potassium pump in action, which is trying to take out the sodium and bringing in the potassium. Then we also talked about, yes, there are these channels, more potassium channels there, which through which potassium could easily diffuse out, right? And the sodium that you're looking at is actually entered because of the electrochemical gradient, because already there's sodium, more sodium outside, right? And there is this environment is more negative. So sodium is actually entering through this electrochemical gradient. But this is the part which is actually the resting axon, right? This is the part when a stimulus has been applied and now depolarization happens. So you can see these voltage gated sodium channels open and sodium is actually coming in. So look at the direction. See, so initially it was more positives outside, more negative inside. And now depolarization, may have a look. There are more positive inside. The situation outside becomes negative. See, so what will happen next? After some time, when it reaches plus 30 millivolts, sodium channels would close, right? And these voltage gated potassium channels would open, right? So this place is now repolarized. So have a look here, right? So what will happen now? These voltage gated potassium channels would open, more potassium would go out. So now you see this environment has become more positive, right? And then you can see it is actually being propagated. Now the next part becomes depolarized. So look at the charges. So this is how this action potential takes place. It's a flow of charges and it goes in one direction only. Okay, now, so let's talk about all or nothing principle in detail. So when these receptors are stimulated, we just discussed they are depolarized, right? But if the stimulus is very weak, remember the resting potential is minus 70 millivolt, right? And the threshold value was minus 55 millivolt. So if the stimulus is very, very weak, the cells are not depolarized very much, right? So a few sodium channels would open, right? But the cells are not depolarized very much. And the sensory neuron is not activated enough to send the impulses, right? Because remember, it was a positive feedback. So more and more sodium channels would open and more sodium ions would flow in. But that would happen only when the stimulus is strong enough. If it is weak enough, right? So it would not be depolarized very much. If the stimulus is stronger, so sensory neuron will be activated. It will transmit impulses to the central nervous system, right? But remember, it has to actually cross this threshold, which is minus 55 millivolts. Only then local depolarization of the receptor cell will happen, right? So if the receptor potential is above this threshold, the receptor cell will stimulate the sensory neuron to send the impulses. And above this threshold, action potentials are initiated in the sensory neurons, right? So this is what we call as all or nothing principle, right? So either they will happen, but they have to happen like above this threshold value, right? Below this, they will never happen, right? So all or nothing means either they will happen, so the stimulus has to be strong enough, or they will not happen at all, right? Or in other words, the neurons would either transmit impulses from one end to another if the stimulus is strong enough, or they would not do that at all. Okay, here, so you're saying, see, this is all or nothing. So if this threshold value is crossed, only then this stimulus actually, this nerve impulse will be generated. Okay, here what you're looking at is that this is like being shown as high frequency of impulses, right? So this is being generated when the stimulus is strong enough. But look at the peak value here. It remains the same at the same value of minus plus 30 millivolts, right? Plus 30 plus 40 millivolts. It stays, it never ever goes above that value, right? But what you notice here between this diagram and this diagram, so high frequency may actually you will be able to see more action potential being generated in the set time, right? But if the stimulus is weak, so you will simply see a weak frequency there, but the height would remain the same. See, this would remain the same. So as the strength of the stimulus increases, the receptor potential also increases. And if the receptive potential reaches a threshold, then impulse is sent along the sensory neuron at low frequency. Increasing the strength of the stimulus above this threshold increases the frequency of the impulse. It does not change their amplitude. So remember, the amplitude remains the same. It's just the number of like waves that you're seeing here are more, right? And no impulses are produced when the receptor is giving a very weak stimulus. So if it doesn't even cross this threshold value, to look at this horizontal line, no impulse actually will be produced at all, right? So this is like, if it helps you to understand all or nothing principle. So this is another diagram. So this is actually from a very strong stimulus, right? And this is a weak stimulus. But if it is even very weak, that if it even doesn't cross the threshold, so there will be no impulse at all, right? So what the only difference is there in the frequency. The amplitude remains the same. So this value is fixed. This plus 30 millivolts is fixed, right? The resting potential at minus 70 millivolts is fixed. Okay then, so this is speed of conduction of impulses actually refers to how quickly the impulse is transmitted along a neuron. And this speed is actually determined by two main factors. The first factor is actually the presence or absence of myelin, right? Because it totally depends on whether or not the axon is insulated by a myelin sheath or not. So this presence or absence of myelin actually determines the speed of conduction. And second is actually the diameter of axon, right? So these are the two main factors, right? Temperature is another factor, but the two main factors are presence or absence of myelin and the diameter of the axon. Okay then, so let's talk about the presence or absence of myelin. So this myelination means presence of myelin, right? So myelination and saltatory conduction. So in unmyelinated neurons, the speed of conduction is slow, right? So there's no myelin, the speed of conduction would be slow. And it will be so slow and so low actually, so as slow as 0.5 meters per second, right? So that would be the speed. But if it is myelinated, so remember they were myelin sheath and then they were like nodes of Ranvier there. So in a myelinated human neuron, the action potential can travel at speeds of up to 100 meters per second. So look at the difference here between unmyelinated and unmyelinated, right? So because of myelin, what is happening, this myelin speeds up the rate at which the action potential travels by insulating the axon membrane, right? Because these sodium ion and potassium ions, they cannot flow actually through the myelin sheath because it's insulated. The part of the axon is insulated, right? So they cannot flow through this myelin sheath. So for that part, actually, it is not possible for depolarization action potentials to generate in those parts of the axon which are actually insulated, right? Because they're surrounded by the myelin sheath. So action potentials cannot be generated there. Action potentials are actually generated around those nodes of Renvier, right? The little gaps that you see between these myelin sheaths, right? So action potentials can only occur at these nodes of Renvier. And this is the area where all the channel proteins and pump proteins are actually concentrated. Got it? So this is actually what we call as saltatory conduction. So that's why we say okay, they jump from one node to the next. So if the like axon is myelinated, right? So the speed would be more, around 100 meters per second, because it would take less time for this nerve impulse to jump from one node to the next, right? So they keep on jumping and going in one direction only. So see here, this is node of Ranvier. This is another node of Ranvier. So see, this part of the axon is actually insulated. So this is a myelin sheath. This is insulated, so action potential would not be generated there, right? So where it is being generated, this part here. This is the part where these voltage gated channels and all these channels are actually concentrated. So this is a very small area. See, so that's the reason it actually seems like to jump from one part to the next, right? So these local circuits actually exist from one node to the next. That's the reason action potential jumps from one node to the next. And for this is a distance of 1 to 3 millimeters. So you can see this distance here, from here to here. So there's a distance of around 1 to 3 millimeter. And this is what we call as saltatory conduction, right? In a myelinated axon, this saltatory conduction can increase the speed of transmission by up to 50 times than that in an unmyelinated axon of the same diameter. Got it? So this word, this saltatory, has actually come from Spanish, like salta in Spanish means to jump, right? So these are known as saltatory conductions. So just see if the channels are open here, the channels here are closed, right? And then look at this movement. So they would actually jump from here, then they would jump here, right? And finally they would reach the synapse, right? So this is what is known as saltatory conduction. Okay, the second factor that I would actually like have an impact on the speed of conduction is the axon diameter, right? So thick axons transmit impulses faster than the thin ones, as the resistance is much less, right? So if the axons are thicker, the resistance is much less, and they would actually transmit the impulses faster, right? So there's an example of earthworm. Earthworms actually have no myelinated axons. So what they have very small number of very thick unmyelinated ones, right? But even though they have no, like there's no myelin, right? But since they're very thick, so and these thick unmyelinated ones, they run all along their body, right? So if there's a bird which is actually pecking at an earthworm to eat it, right? And what would happen? This would actually act as a stimulus. It would help in setting up these impulses along these giant axons, and they would travel very, very quickly because this axon is very, very thick, right? It is resistant, it is thick, so it will stimulate the muscles to contract, right? So this rapid response helps the earthworm actually to escape from that bird, right? So the reason is these thicker axons have an axon membrane with a greater surface area over which diffusion of ions can occur. See, so this increases the rate of diffusion of sodium ions and potassium ions, which in turn increases the rate at which depolarization and action potentials can occur, right? So that's the reason these thicker axons actually are helping in transmitting the impulses faster, right? The reason is they are thicker, so it means they have a great surface area over which diffusion can occur, right? And we can then be rest assured that the rate of diffusion of sodium potassium ions will be increased, right? And this is through which the rate of depolarization action potentials would actually occur also. The third reason is temperature, right? So the reason is this temperature, if it is more, ions will diffuse faster, right? So hence the speed of conduction will actually increase when the temperature is more, right? But like we need to take care of the fact that denaturation of proteins also occur at around 40 degrees Celsius, right? Then after that, the speed would begin to decrease, right? So that's another point. So there are two main factors actually that contribute to the speed. So first is the myelination, second is axon diameter, and third is the temperature. But definitely, like it can happen below 40 degrees Celsius, but if the temperature actually goes past it, so proteins will actually denature, right? So speed would then slow down. So I hope you have got the idea about how an action potential is generated. So let's watch a video to review this concept. And in the next video, we will talk about the synapses.
Neurons communicate with each other through their dendrites and axon. Generally, incoming signals are received at dendrites, while outgoing signals travel along the axon to the nerve terminal. In order to achieve rapid communication over its long axon, the neuron sends electrical signals from the cell's body to the nerve terminal along the axon. These are known as nerve impulses, or action potentials. An action potential is essentially a brief reversal of electric polarity across the cell membrane. Cells are polarized, meaning there is an electrical voltage across the cell membrane. In a resting neuron, the typical voltage, known as the resting membrane potential, is about negative 70 millivolts. The negative value means the cell is more negative on the inside. At this resting state, there are concentration gradients of sodium and potassium across the cell membrane. More sodium outside the cell and more potassium inside the cell. These gradients are maintained by the sodium-potassium pump, which constantly brings potassium in and pumps sodium out of the cell. A neuron is typically stimulated at dendrites, and the signal spread through the soma. Excitatory signals at dendrites open ligand-gated sodium channels and allow sodium to flow into the cell. This neutralizes some of the negative charge inside the cell and makes the membrane voltage less negative. This is known as depolarization, as the cell membrane becomes less polarized. The influx of sodium diffuses inside the neuron and produces a current that travels towards the axon hillock. If the summation of all input signals is excitatory and is strong enough when it reaches the axon hillock, an action potential is generated and travels down the axon to the nerve terminal. The axon hillock is also known as the cell's trigger zone, as this is where action potentials usually start. This is because action potentials are produced by voltage-gated ion channels that are most concentrated at the axon hillock. Voltage-gated ion channels are passageways for ions in and out of the cell, and as their name suggests, are regulated by membrane voltage. They open at some values of the membrane potential and close at others. For an action potential to be generated, the signal must be strong enough to bring the membrane voltage to a critical value called the threshold, typically about negative 55 millivolts. This is the minimum required to open voltage-gated ion channels. At threshold, sodium channels open quickly. Potassium channels also open, but do so more slowly. The initial effect is therefore due to sodium influx. As sodium ions rush into the cell, the inside of the cell becomes more positive, and this further depolarizes the cell membrane. The increasing voltage in turn causes even more sodium channels to open. This positive feedback continues until all the sodium channels are open and corresponds to the rising phase of the action potential. Note that the polarity across the cell membrane is now reversed. As the action potential nears its peak, sodium channels begin to close. By this time, the slow potassium channels are fully open. Potassium ions rush out of the cell, and the voltage quickly returns to its original resting value. This corresponds to the falling phase of the action potential. Note that sodium and potassium have now switched places across the membrane. As the potassium gates are also slow to close, potassium continues to leave the cell a little longer, resulting in a negative overshoot called hyperpolarization. The resting membrane potential is then slowly restored thanks to diffusion and the sodium-potassium pump. During and shortly after an action potential is generated, it is impossible or very difficult to stimulate that part of the membrane to fire again. This is known as the refractory period. The refractory period is divided into absolute refractory and relative refractory. The absolute refractory period lasts from the start of an action potential to the point the voltage first returns to the resting membrane value. During this time, the sodium channels are open and subsequently inactivated while closing and thus unable to respond to any new stimulation. The relative refractory period lasts until the end of hyperpolarization. During this time, some of the potassium channels are still open, making it difficult for the membrane to depolarize, and a much stronger signal is required to induce a new response. During an action potential, the sodium influx at a point on the axon spreads along the axon, depolarizing the adjacent patch of the membrane, generating a similar action potential in it. The sodium currents diffuse in both directions on the axon, but the refractory properties of ion channels ensure that action potential propagates only in one direction. This is because only the unfired patch of the axon can respond with an action potential. The part that has just fired is unresponsive until the action potential is safely out of range. An action potential generated at the axon hillock usually travels down the axon to the nerve terminal and not back to the cell body. This is because the concentrations of voltage-gated ion channels are higher in the axon than in the cell body.